Everyone, and welcome to our September YouTube live Q&A from the Kidney Project. I'm Liz Grass. I'm our Director of Partnerships, Communications, and Regulatory Affairs for the Kidney Project, and I'm excited to be talking with Dr. Shuvo Roy today, who is the Technical Director of the project. And we will be diving deep into the development timeline for the implantable bioartificial kidney. So we'll spend about the first half of the hour with Dr. Roy presenting the clinical specifications, the milestones that the device has met to date and what is still ahead. And then in the second half, we'll get to the audience questions that were submitted on the registration and as well as from the chat. So just one reminder, if you're not subscribed to our YouTube channel already, please subscribe if you want to participate in the chat, and we'll get going. So yeah, thanks again for being here. And Dr. Roy, you want to get started?
Yes, welcome everybody, and thank you for this opportunity to speak with all of you again. It's been fantastic to get all the feedback we've gotten because of these YouTube live chats, and also thanks for your comments on social media, as well as your emails. So the topic today, based on audience interest, is really about how we move forward on the implantable bioartificial kidney and move it into patients and make it a product available commercially. It's a big task. We'll try to do the best we can. But I thought I would start off by doing what I normally do, which is remind for those of you who are not here all the time about what is our ultimate goal, which is building an implantable artificial kidney. And some of you have seen the video, but I'll play the video just to get everybody back on levels here. So I'm going to share my screen, and you'll see a blank screen, and I'll share my sound and we'll play the video. And then I'll pick up on the other side of the video.
People living with kidney failure can soon look forward to a future without dialysis or an endless wait for a transplant organ. Combining continuous treatment with total mobility, the implantable bioartificial kidney will give patients back their health, freedom, and quality of life. The compact device will replicate many functions of healthy kidneys and will not require immunosuppression drugs. The bioartificial kidney combines a mechanical hemofilter to remove toxins from blood and a bioreactor containing engineered renal tubule cells to maintain water volume, electrolyte balance, and metabolic functions. Highly efficient membranes constructed from semiconductor silicon wafers enable filtration without requiring pumps or electrical power, while protecting the renal cells from rejection by the patient's immune system. The biocompatible device attaches to the circulatory system and removes toxins to the bladder as waste. After a decade of engineering development to refine the hemofilter and bioreactor components, preclinical prototypes of the implantable bioartificial kidney have been successfully created. Cleanroom microfabrication processes have been established to reliably produce high-quality silicon membranes. Tissue culture protocols have also been optimized to reproducibly grow renal tubule cells and maintain their function. The silicon membranes were mounted in polycarbonate housing to construct hemofilters, and in similar fashion, bioreactors were assembled to immunoprotect the renal tubule cells. The hemofilter and bioreactor components were then connected to create bioartificial kidney prototypes for benchtop and preclinical testing. In proof-of-concept experiments, the hemofilter operated under blood pressure alone, without systemic anticoagulation, while the renal cells in the bioreactor remained alive and healthy without immunosuppression. Further improvements will scale up the prototypes with additional silicon membranes and renal cells, creating clinical devices that can demonstrate sustained treatment of kidney failure in human trials. The implantable bioartificial kidney will provide the therapy and mobility patients need to move beyond the burdens of dialysis and organ shortage, and once again enjoy the freedoms of everyday life.
All right, so now you've been refreshed with our goal and the progress we have made over the last decade. I thought as we talk about, breaking down the timeline, which is a topic of this YouTube, it's instructive to see for me and for all of you just where we started and where we've come. And for that, I put together a few slides that I'll share with you, and you'll get a sense for the work that has been done and needs to be done and how we hope that will be accomplished. So let's do another share. You probably see this, so I'll start off here. So some of you online have seen a version of this graphic many times. And it's a graphic I use over and over again because it's something that we developed very early on to guide our mission. And we try not to stray away from what our ultimate goal is, which is an implanted device that functions as a kidney. And it provides continuous treatment, allows the patient to be mobile. And because of how it's structured, a filtration unit, which we call the hemofilter, that mimics our kidneys filtration unit, the glomerulus, couple that with a cell therapy unit, which we call the bioreactor, which also functions as a kidneys tubule. By putting them together, you get something that will allow us to achieve the benefits, or the vast majority of benefits of a kidney transplant, including the ability to have a more liberalized diet. Drink what you want to a certain extent, eat what you want, again, to a certain extent, but much more freedom than you get with traditional regimens of dialysis diets. And this has been our approach. You'll see basically the two units, the filter or hemofilter, and the bioreactor, or sometimes I'll call it the cell therapy unit. And of course, it can be sensorized, so we can transmit data on the status of the device as well as that of the patient. So many of you want to know, well, how do we move forward from here to something that goes into patients? And what I'll share with you today are the details on how we've been moving this forward and what we still have to do. But one thing I do want to bring up is that we are not the first bioartificial kidney, and we're building on the shoulders of giants. And this is the work done almost 2 decades ago, starting at the University of Michigan. And here's a pioneer of the bioartificial kidney. And it does not necessarily use new technologies per se, in the sense that I will talk about in a few minutes, but it tested out the concept of filtration and cell therapy to mimic our own kidneys architecture. So Dr. David Humes, a nephrologist, treats patients who are in the ICU with kidney failure. Often this is because of sepsis. It may be because of trauma, could be because of, drug overdose, etc. And the standard treatment has always been for these patients intensive dialysis as sort of the last resort. And often just the intensive dialysis did not work. So he hypothesized, maybe if I provide some cell therapy in conjunction with toxin removal, there might be a benefit. And indeed, using the existing technology available at the time, which you see on the right, he was able to take standard dialyzer cartridges, cadaver kidney cells, put them into the bioreactor, and he showed that through a series of preclinical and clinical studies that it is possible to reduce the deaths. Basically showed that this actually had a survival benefit for patients who would have otherwise died. So for us, and my colleague, Dr. William Fizel, who many of you have heard from and seen, latched onto the idea that here was an approach to building a treatment that mimics the natural kidney, the native kidney. And that's exactly what was done and showed a clinical benefit. So for us, we are building on a significant amount of work that's already been done. We're not trying to discover new principles of kidney function, but rather, can we take this approach and advance it? And what was attractive to us about Dr. Humes' work was that he was taking biological and synthetic combination. What I show here on this slide is the native kidney tissue, the glomerulus, which is the filter, and the tubule that provides the metabolic functions of a kidney. And what Dr. Humes did was basically use the first cartridge as a filter, and I'm calling it the artificial, a synthetic filter, just a polymer hollow fiber dialyzer. In the second, he took a filter, but the real working component was kidney cells. He was able to harvest from a cadaver kidney that could not get transplanted. And so basically he was taking the approach of, let me combine an artificial filter with biology of kidney cells to provide more function than dialysis does and closer to what a kidney transplant is. So in other words, he was constructing a mechanical filter and then growing a biological component and putting them together. And the attraction of the biohybrid, I think it takes the lessons learned from both biology and membrane technology, brings them together, and in some ways accelerates the pathway to getting to patients, at least on the scientific and engineering path. So for us, this has been basically the mission of the Kidney Project, is take the work that was demonstrated in the ICU to treat acute kidney injury and make it available for the end-stage renal disease community, the people who do chronic dialysis, and make something that builds on the acute kidney injury renal assist device to an implantable bioartificial kidney we call the iBAK. So that is the pathway that we defined over a decade ago. And we then started laying out a development plan of how we move forward. But it should be instructive for us to think about, how do you move forward? What are the enabling technologies? And to do that, we should first look at the features of the iBAK. What makes the iBAK stand out in terms of its features, which will also drive what technologies need to bring into the system so we can make it work, are the fact that we have a hemofilter and a bioreactor. So we want to make something that just runs off blood pressure alone and does not require electromechanical pumps. Why is that? Because that lets us make something that's compact and small. If we can do that, we also take away the failure that may be associated with batteries running out or the pump mechanically failing. So we desired that we want a system that does not require electrical power and does not require energy-intensive and bulky pumps. We also want a filter that mimics the kidney's membranes. And that, basically in engineering speak, means it should have a high permeability, means it can filter a lot of toxins with a small surface area with little driving pressure, as our kidneys do, but it should not leak any albumin or any other desirable molecules. So we call that selectivity. So the membrane has to have high permeability, high selectivity, and then to minimize the likelihood of infections, but also encumbrances with being connected to something outside. We said it should be totally contained. And if you're going to put cells here, can we avoid the need for immune suppression drugs? Because those have their own side effects. And then we also know that anytime you put an artificial material in the body, especially with contact with blood, it may clot. So can we actually make the device operate with blood in a friendly manner, but do not use anticoagulation drugs, the ones like warfarin and Coumadin that have their own harsh side effects? And the whole device should be biocompatible. So it should basically behave as a permanent implant, but recognize as engineers that if something fails, we can go and attempt to repair it or replace it. So these are the features of the iBAK. Again, to get compact size, no electrical power for batteries, avoid or large bulky pumps that also draw a lot of energy, high selectivity and high permeability membranes that allow for a lot of ultrafiltration, removal of toxins, that is, as well as no leakage of albumin or anything that's desirable, no wires or tubes going outside, and then no need for immune suppression drugs or anticoagulation drugs. Certainly minimize, if completely not eliminate them. And then we should have long lifetime, it should biocompatible. And the device should take the blood from the artery, pass it into the device, process it. The urine is directed into the bladder, and the processed blood is sent back into the vein, very much like a kidney transplant. So using that as our desired features, we said, okay, let's come up with a development timeline. And the key technology that we use to achieve a compact device with the performance features that we want is this artificial technology, which I call the silicon nanopore membrane. It has high permeability, it's highly selective, it's biocompatible. That's work we've done over the decade. And we've shown that we can actually make a membrane that meets a lot of these specs that are here. And because they have high permeability and high selectivity, it also means they use a lot less driving energy, a lot less surface area, so we can use the blood pressure alone. And it's biocompatible, so it doesn't clot and does not necessarily get gunked up by proteins in blood. But this is the artificial membrane technology. And then we also bring biology for the iBAK. We decide that we're going to use kidney cells, very much harvested, again, in this case, from a kidney cadaver, or rather cadaver whose kidney could not be transplanted. But we're also looking at other cells, animal cells, and eventually we hope that we'll find stem cell sources for this. But for our current work, we are driving the biology using kidney tubule cells, and we've shown that they can actually do what they do in nature, in our native kidneys, which is they should be able to transport water and salts, different types of salts, sodium being the most common, but there are other salts, calcium, magnesium, etc. And they should be maintaining their function for a long period of time. So we call that sustained phenotype. It basically means, are these cells behaving as they should and performing the function that they should? So our work, and those, especially that of my colleague at Vanderbilt, they have carried these cells out to almost 10 months. Think about that, in a bioreactor construct, in vitro, in the lab environment, that the cells are functional for many, many months at a time. And they, we feel like it should, and they should be able to continue performing, but most lab experiments, for those of you who know, just run for a few weeks at most. So we are very excited that we actually have a filter that I described in the past slide that works as it does, and cells that function, but also function for a long, long time. So I'm going to summarize all the progress we've made over the last decade. On the left-hand side is the progress that is related to the filter. On the right is the progress related to cell therapy. And what I'm showing here is that we have shown that we can make the silicon membranes and show pump-free hemofiltration, meaning no electrical pumps, just blood pressure alone. We can also design the path of the blood flow through the device so that it does not cause turbulence, which can lead to poor function, but also can lead to clots. We've been working on trying to scale up the device from animal scale to human scale. We've made prototypes of the human scale, and we know that it's possible to do it, but we need to make, and I'll talk a little bit about the development pathway, but going forward, we know it is possible to make clinical scale devices. We have coatings that actually prevent blood clots or just deposition of proteins from blood that will gunk up the membrane. And then working with a team of talented surgeons, we show that we can actually implant this in preclinical models, animals, and that this device is accepted in by these animals and do not cause adverse reactions for a short period of time, which is the length of our studies to date. And also, obviously, for the filter, we have to make sure that it doesn't get affected by sterilization techniques. Sterilization is a very important part of anything that goes inside the body. So we want to make sure that if we have coatings, we have the silicon membranes, the different sterilization techniques do not degrade their performance. And we verified that we have a series of sterilization techniques that are available to us if we want to sterilize this filter. For the cell therapy unit, we've shown that we can actually harvest kidney cells from cadaver kidneys and preserve them in liquid nitrogen and use them sometimes as much as a year later. We've also shown that these cells exhibit the function outside of the body as they do inside the body. And that's what that second line means: inhibitable active transport of proximal tubule cells. What basically saying that these cells behave as our kidney's proximal tubule cells in an artificial construct as they do in a native kidney. And we've been able to test that, as I've mentioned, for up to 10 months. The third one I already hit on is about preserving the cells in long-term storage. Why? Because for our experiments and our development, sometimes we'll need to keep the cells so we can use them when we need them. Otherwise, they'll go bad. And I think if you think in terms of future, that's something we'd want to know, is how do you preserve the cells that you can use in device construction later. Also importantly, we've shown that the membranes can protect the implanted cells without immune suppression drugs. What does that mean? Basically means that we can put the cells on one side of the membrane, expose the membrane to blood or a solution that has a whole bunch of molecules that destroy cells that are associated with immune reaction. And we've shown that the membrane itself, because of the pores we have in the membrane, allow nutrients, oxygen, glucose, sugars, other sugars, very small proteins to go through, but do not let the cells and the big molecules that cause rejection to get from one side of the membrane to another. Basically saying that, yes, we have a way to think about providing a bioreactor that does not require immune suppression drugs to function. My colleagues at Vanderbilt have been also working on, can we enhance the cell operations so that you actually get more transport? In our kidneys, we have square meters, I'm using scientific units here, of surface area. If you try to pack all that into a device, it could become a large device. So the kidney really compacts them. But in our case, we said, can we actually use a less number of cells, but enhance their function by using gene editing technologies? And we're exploring that, and we've shown success with that. And then can we continue to optimize the culture conditions, the conditions how the cells are grown, to make sure the phenotype is sustained, meaning they'll function as long as possible. And we've shown that only 10 months, but we continue to optimize this because we want to make sure that this can work as long as we can and also enhance transport. So this is the milestones we've achieved, individually for the hemofilter and individually for the bioreactor. So when we achieved this, this was right around the beginning of the pandemic. Everything slowed down, but we continued working. Luckily, we are able to somehow go at a very slow pace, but we took this knowledge that we gained from the hemofilter and the bioreactor and said, okay, can we put something together that basically recapitulates what I showed in that first slide, the vision slide of the bioartificial kidney. So this is our attempt, a CAD drawing, that shows how we're going to put together a prototype hemofilter and a prototype bioreactor into a single functional prototype of a bioartificial kidney. So this is an exploded diagram. And here's actually the first functional prototype. This is real, where we've actually got membranes in the device, in one case for filtration of blood. And at the bottom, we have membranes that protect the kidney cells. And this is what you saw in the video. We took this and put it in our preclinical models to see if it will function. And we're very, very excited to see that it functioned, it worked, it produced urine. It produced very little urine, not because it was not working, it's because we had very limited amounts of surface area and cells. And really the goal was, can we actually create urine by combining a hemofilter and bioreactor without immune suppression, without anti-clotting drugs? And the answer was yes. So we have a small-scale device that we've achieved. Now we're going to look at how do we move forward. So we have a small scale device that produces urine. We need not to build clinical scale devices. What do I mean by that? That device had about 100th the surface area of what we need. Why is it 100? Because that's the machining technologies we had, what was available in the pandemic. We put together things, but we want to now put in more silicon membranes that are optimized for higher filtration capacity, pack in more kidney cells. So to do that, we have to build clinical scale devices. Those are animal scale prototypes, and they would not provide enough therapy to keep a sick patient alive. So we need to build clinical scale device. We know what the designs are, but we're going to make it at a larger scale and a bigger size. We also want to continue to optimize the cells so that we can make sure the cells survive longer and prove to ourselves that there's going to be no adverse reactions, which we think there will not be, but we want to continue optimizing it so that we can pack more cells. So we pack, again, a few cells, tens of millions of cells. We want to pack hundreds of millions of cells. So again, to do that, when you're packing so much, sometimes you have to optimize the conditions, the geometry, so that the cells will maintain their function. As we build these clinical scale devices and optimize cells, we're going to think about bringing together this device. This is a small scale device, but basically integrating clinical scale hemofilters with clinical scale bioreactors into a single clinical scale implantable bioartificial kidney. What is the work to be done? You got to build the devices independently, bring them together. That's what I call device integration. By itself, okay, now you've got a device that can provide sufficient therapy, but you have to still show that it works and keeps somebody that with that kidney function alive. And what I mean by this, someone, first you want to test it in a preclinical model, an animal that does not have kidney function, and show that our device will actually keep the sick animal alive without the need for dialysis. And that data that we get is going to become the basis for what we'll submit to the FDA for the first human study, which we're calling the pilot study. So in this timeline, what you're seeing are the key work that has to be done. And most of it, you'll notice, is engineering, manufacturing type stuff. Then in the iBAK demonstration, that's all that you do. First do the testing, you get the data, show for toxicity, show for biocompatibility, create the package that goes to the FDA. They'll review it. Assuming there's no unanticipated issues, they would get approval, and then we could do the first human study of the iBAK. Now, I've mentioned this before. My colleague has mentioned this before. This is mostly an engineering effort. It's not a discovery science effort. We know how to put the thing together. We know what the filter should look like. We know what the cells perform or how they can be made better. We want to continue doing that, but we need to put it together, and that takes resources and takes time. But if everything goes well on both fronts, it is conceivable that within 3 years, you're able to build a clinical scale device and test it in a preclinical model and apply to the FDA for the first human study. There's some ifs there, absolutely, but we feel confident with the team we have, if appropriately resourced, and again, forbid another pandemic or anything like that, it's possible to get to this within the 3 to 4 year time frame. Now, this is the first patients. There's still work to be done. We have to work on making this commercially viable, getting partners, and those are all important. And those will take partners beyond us, but at least we think that after you go to the first pilot human study and you look at what's been done for heart valves, insulin therapy, it's another few years that before it becomes a commercial product. So I think it's conceivable that we can think that within the next 10 years or so, if we're able to get the resources and the team put together to do this and the partners, this could become a commercial product out there. So I'll end there. And that's sort of a very high level way of thinking about the iBAK development timeline and what work has to be done. And I'm happy to answer questions that you guys are sending in via the chat.
Great. Thank you so much, Dr. Roy. That was very informative, and we have a lot of questions coming in. I'll ask you one that just came in. Would it be possible for a patient eventually, once iBAK is done and approved, who is on dialysis because of multiple myeloma, to get the iBAK?
So basically the question is, can kidney cancer patients, or any cancer, not kidney, but kidney cancer, but can cancer patients get the iBAK? I'm going to give you my answer, not as a clinician, because I'm not, and I also don't know your personal situation, but I'll give you the answer that I know based on what I know of the device design, device results to date, and what I've heard from my colleagues who are nephrologists and transplant surgeons. The device is self-contained, and it will be implanted, and you'll be able to get filtration and the cell therapy. Now, remember, if the body's immune system and the other systems cannot attack the cells, then the device continues to function. So in principle, I'd like to say and think that, yes, that a similar situation like yours would be potentially could use the artificial kidney, but of course that will depend on very many factors, including yours or whoever's health at the time for surgery.
Great, thank you. The next question may be obvious to a lot of people, but it still comes up. Is the device available now for patients? Can anyone get the iBAK now?
No, thank you. That's a great question. We do get it, despite thinking that we answer it all the time. The short answer is no, it's not available to the public. It's not available at the clinical trial stage. We have done the work to show that it's feasible in technology and in preclinical models, but we're not at human studies yet. And the last slide I showed showed the timeline and to the first clinical study, and then the work that will be needed to get as a commercial product. So we are hopeful. It's going to require all of us to work as a team to get it out there, but right now it's just still in the preclinical stage.
Thank you. And from the point where we begin clinical trials, how long do you estimate the clinical trial process to go on until the device is fully approved?
Yeah, another great question, guys. And I think thank you for asking. So I think the way to answer that question is to look at devices of comparable complexity, or even device that'll inform us. Where do we look to? So we are a device, first and foremost. We're not a pill. So let's look in the field of devices. So look at new types of stents, heart valves, brain stimulators, artificial heart. And when you look at them, what happens? Typically, you show the proof of concept data, you get to the first clinical trial, and then usually the first clinical trial is all about establishing safety. That's primary for the FDA, and it's also important for us, right? It's very important. So we want to make sure it's safe. Once you've established safety, then the device has to go for a pivotal study. So there's a pilot study for safety and a pivotal study to gather statistically significant data that shows your device is both safe and effective. So that requires to do many more patients. You have to have many more patients on the device. It should be done at more than one place and by more than one surgeon or nephrologist monitoring them, and different types of patients across different health conditions, genders, and etc. So how long does that take? So I'm going to look at the work that is done in artificial hearts, stents, TAVR. Sometimes that work goes as fast as 3 to 4 years. Sometimes it can stretch out beyond that. But so if we take the best case scenario, you have to do a clinical study. Some people hear the term randomized control studies. That's another way to think about it. Those RCTs, randomized control trials, they take usually a few years. So if I was to give you a sense, best case scenario, probably you have to think about making more devices, finding the partners, getting ready for the clinical studies for another 4 years. So I think when I think about it, another 3 to 4, 5 years is not unreasonable. What is unreasonable is probably, is it going to be done in a year after you do the first human study? I don't think that's practical. Is it going to take 20 years? Not because of the technology, maybe because of resources, but not because of the technology available. But if we had the technology available and resources available, then it's probably 4 or 5 years is not an unreasonable time frame.
Okay, thank you. And then that's for maybe the US approvals. What about around the world? I think we have a lot of people from different countries tuning in, and they're all interested to know about when it might be available in different places.
Yeah, so obviously as the engineering lead on this project and technical director of the kidney project, we are not limiting ourselves to the United States. And the reason we do a lot of the work here in the United States because this is where we are. We have the access to the team and resources. But again, going back to when it will be available internationally, from our perspective, it will be available as soon as we find partners in these countries that can manufacture. If not manufactured, it's distributed for us. So it could be manufactured in one country and distributed by another. So it's going to require partners. So let's say you're in a different country, has been approved in the US. Let's look to other devices that have already been approved. So I look at pacemakers. Pacemakers may be developed here. When is it available in a country, say in Asia, in Africa? Well, it depends on the specific country. Some countries have their own version of the FDA that you must get approval or clearance authorized by. So you have to submit the data to them and say, okay, here's the data, and that agency in that company is going to say, okay, great. There are some countries that will say, oh, you got approved in the US and a few other countries. Okay, just share with us the data, and it's just more. You don't have to do any more trials here. You move into the market. So it's going to be very country specific. But in general, I would say after it's approved in the US, from our side, if we have the partners, we want to move it right away. So it's now there are large companies sometimes that think about, hey, we're going to have a simultaneous approval. Those things are very hard to plan out ahead. But I think my answer to you guys who are asking that question is, are there partners in your country that are interested? They should start thinking about how they might join with us well ahead of time, so that when we get to that point, they're ready to carry the baton, so to speak, and move it into those markets.
Great, thank you. There's another question about will patients that have the iBAK transplanted into them need immunosuppression?
Yes, so many of you know that the iBAK is supposed to function like a kidney transplant, and because it's got both cell therapy and filtration, mechanical filtration, and it's supposed to be totally implanted and allow the person to be mobile, eat and drink what they want. And that sounds a lot like transplant. So I'll put out 2 points here. One is we are going to provide many of those same functions of a transplant, and we're really focused at providing mobility and ability to have more liberalized diets, and those hopefully lead to better health and quality of life. So will you need immunosuppression? The short answer, we hope not, and we think not. So when we put together our cartoon, we talked to lots of people, and we found out that one of the reasons kidney transplants fail is because the drugs the patients are given to avoid rejection can also be nephrotoxic. They basically poison the transplant kidney. So over time, the transplanted kidney gets adversely affected by these immune suppression drugs and lose function, such that the average time of a transplant, life of a transplant, is about 10 to 12 years. Of course, some people longer, some people shorter, but that's the average. So if you have to avoid the need for immune suppression drugs, it would last longer. And because our kidney cells are inside the bioreactor, which is basically surrounded on the outside by the silicon nanopore membranes, which have holes small enough to let toxins and nutrients pass through, but do not let the immune components go through. We do not anticipate that we'll need immune suppression drugs. And we've shown this. We've done the bench studies to show the molecules don't pass through. But more importantly, we did that animal study. That animal was not an immune suppression drugs. We took the device apart, and that was what the video showed in green. The cells were still alive and functional. And in other studies where you have just foreign cells injected into another body, animal or not, they immediately get attacked and die. We didn't see that. In fact, we had almost close to 95 or 100% viability, meaning the cells were alive and functioning. So we are optimistic.
That's great to hear. Switching gears a little bit, do we have any estimates for how much the device might cost once it's available?
Gosh, good questions, guys. Write the checks. I think we are at the stage where we are developing the device. So I think there's a certain price to device development, but what a device costs is not a function of what a device costs for the person purchasing it is not necessarily what it costs to make. And we should be aware of that. There's a medical device industry. There's a whole economics around device development that goes in. So you might say the device costs X to make, but they charge many multiples of X when they sell it. And they're just business justifications sometimes why that happens. But in general, I think in my mind, if I sort of project the cost of goods, what does it take to actually build it? And what one might sell it for? I'm again going to use some of the averages I've seen in the medical industry. And that's how I think a business might eventually do this and still be competitive. I would envision that it should be less, it would cost less than dialysis for the insurance company. Because if it costs more than dialysis, you really have to justify it. It gives you benefits, you have to justify it. But I'd like to think that it could cost less than dialysis, probably about comparable to transplant as it costs today, or even maybe less because you don't need immune suppression drugs. So you're asking, what are those numbers? Well, today in the US, dialysis costs Medicare, the primary payment institution, about $90,000 per patient per year. Think about that. $90,000 to do dialysis in the US for one person every year. Transplant, after the initial surgery, costs Medicare about $30,000. The surgery itself is tens of thousands, but after that, it's just the drugs, often tens of thousands. And the Medicare gives a number of about $35,000. So I know, just based on what I know about manufacturing, that the cost of goods is going to be nowhere close to such a high level. So I'm assuming that will be competitively priced about what it costs for maintain a transplant patient. That's the best I can do. But again, it's going to be a question that's a business question, and there's the questions of supply and demand that you're no doubt familiar with. But we think it will save costs, even by providing something that's maybe less expensive than dialysis. It will save costs, but improve health, and at the same time allow us to make it available to more people that need it. That's one of the challenges of transplant. There's a big inequity. A lot of people just don't have access to it. Many of the other countries, there's inequity around dialysis. So we'd like to make this available to anybody and everybody that needs it, such that you need it, you pick it up from the shelf and make it available.
Great. There's a question about fundraising, and specifically if there's a role for corporations to help with fundraising or to support the development.
So as many of you guys have heard from our prior talks, fundraising, funding is a key gap in why this project hasn't moved faster. And we also had the pandemic. I shouldn't discount that, but fundraising has been a challenge even before the pandemic. We've relied on grants, would love partners, would love corporate partners if they're willing, and they can be different levels. They could be partnering us by giving us money so we can purchase, or we can employ people who will work on the project, or they could provide services that will help us scale up. You saw the manufacturing we need to do, make clinical scale devices, do clinical trials. Those, if we had corporate partners that sort of took that on, not only do they bring expertise, but they also help us take some of the financial load off us. That's very helpful. Then some of you have asked, can corporations support? So that's one, well, obviously two ways. They can give us money directly as sponsors, or they can give us services or provide services for us to offload the financial burden. But the other one is just branding. I mean, many of you who are working in companies, most companies want to do good as well as make money. And if they have a campaign to donate to a cause or raise funding for a cause or associate their company with a particular cause, please think of us. We need that kind of support. Every dollar helps. I know we talk about the large numbers and stuff, but every dollar helps. If your company gets together and puts together an internal fundraise and say, we're going to raise as a group for the kidney project, fantastic. If you guys want to convince your investor group, so investment board, to take an opportunity here, absolutely. If you want to do a charitable giving and we are the recipients, we'd appreciate that. If nothing else, talk about what we do in your company newsletters and company press. Also, if the company does something that can help us advance the technology development into scale up and production, absolutely. So yes, there's a big role for companies, and we'd welcome ways to explore working with companies.
Great. On a somewhat related note, do you anticipate any opposition from big companies to develop this, given that it maybe could hurt the business of existing companies?
No, we get asked that question, and the short answer is maybe, maybe not. I mean, opposition in some ways, maybe by not giving us any money, maybe that's the opposition. Okay. I have not, even the people that you might think in big dialysis companies that are like, well, they don't, you're competing against them. We have people we know there. We have friends who have gone to work for these companies. I don't think anybody has come and said, hey guys, we want you to fail. It's like, well, it's great if you succeed. So maybe the way if they oppose us, and nobody has told me that they oppose us directly, is maybe by doing nothing. So in medical device development, in other products, and having been an engineer that has commercialized cardiovascular products, have worked with companies in orthopedic products. What I know is, companies that are already in the market sometimes get the early stage innovators like myself in a university to say, hey, let's work on this together, and we'll license it to our company, we'll move it forward. We don't have so much of that in the kidney space. So maybe that's the one way I think is opposition, but nobody has come to me and say, look, we want you to fail. And if they do, that's unstated. But I think most important is, that they also recognize that this is a big need. And I'd like to think that the people there also care about the patients and what's good for society. And I know the people that I know there, they do.
Great. There's a question about our iHemo technology and what the timeline to patients for that might be in comparison to the iBAK.
Sure. So for those of you who are not familiar, iHemo is a spin-off product using the same technology we use in the bioartificial kidney. So remember, the bioartificial kidney is part mechanical filtration with silicon membranes, part kidney cells to provide the other functions of a kidney, is totally implanted, creates urine. The filter technology itself is, as you remember from my minutes, a few minutes ago, is highly efficient. It works without a blood pump. It's biologically friendly. We've taken that technology to make a better dialyzer cartridge. But in the iHemo, what we are saying is that let us still implant the dialyzer cartridge, the filter, and attach it to the blood vessels. But on the other side, you have catheters get tunneled out that can connect to an external dialysate pump. And for those of you who may not know this, we'll put out a link so you can see the video of what the iHemo system is, so you can know what we're talking about. And because it uses the same technology for the filtration component as the bioartificial kidney, it avoids all the complexities of cells. So in principle, that should be something that gets to faster to patients, and we are hoping that we can make a more user-friendly home hemodialysis system, iHemo, implanted hemodialysis, that does not require needles. It does not have any blood come outside the body, and you can do dialysis at home safely and conveniently and for as long as you want. And because of the interest in home hemodialysis, we hope it can get to the market faster. Would love to get investors who will take that from us and move it towards a product. So in principle, it should get to a product much faster because they don't have to worry about the complexities of cell manufacturing. And because home hemodialysis, as there's growing interest in that space, we are hoping that investor or company will take that on from us and move it into a product. So it can, in principle, get to patients within 5 years if we have the right partner that can move it. And the artificial kidney would probably be closer to to become a commercial product, whereas the artificial kidney would be closer to 10 years for a commercial product. Again, gross generalizations, but that's one way to think about it.
Good. Thank you. There's a question from someone who I think has probably been following us for a long time. And they say that prior to the pandemic, we had reported that we had applied to the FDA for an IDE. Curious to know the response and reaction.
Sure. So, yeah, I think you have been following the project for a long time. And so it was actually an IDE. IND, IID are similar. IND is usually for drugs, while IDE is for devices. But nonetheless, let me just give a little bit of background. So early on, when we started developing this concept, we are fortunate enough to partner with the FDA under something called the Innovation Pathway Program that has become now known as the Breakthrough Device Program, which we are part. It lets us talk to the FDA and think about all the milestones that you have to hit before they approve you for clinical studies and ultimately a product. So when we started those discussions, we laid out a pathway of how do you get to the first clinical trial. And the questioner is basically saying he or she had heard about it prior to the pandemic, and that is true. So we actually went to the FDA, got a pathway, or got a list of things we have to do, and then we went to our institutional review board and saying, okay, we've talked to the FDA, here are the milestones we have to meet, and what are the criteria we need to do the first human study at our institutions, either UCSF or Vanderbilt. So our ethics board, the institutional review board, they look at ethics. They said, well, we think you should probably do some studies that basically confirm to us that the FDA is comfortable with the safety of the device. So we went back to the FDA and said, look, we obviously think the device is safe, but they want this official determination. And to do that, we'll have to go through your process. And they said, yes, absolutely. We are happy to work with you, but you have to do these additional tests that are done before we can authorize you for the IDE. So this is how we get to the pre-pending of how we get to the IDE. So we started down the path of the work that's needed to get the data. We have to do some biocompatibility toxicity testing. We have to do some animal studies, put that data together, submit to the FDA. We have to make devices. So we made devices, a number of devices, and then we said, okay, let's start the path. And as we were getting started, just around then, the pandemic hit. And that was really disappointing because we had everybody lined up. We had our components. We had a plan. What did that mean? Well, first of all, you couldn't just go into the facilities. So For a long time, if you remember, for those of you who remember the pandemic, you couldn't really go anywhere. Just social distancing, very strict. Everything was shut down. So we couldn't do that experiment. So slowed us down there. Then after some few months, they said, okay, you can go in. And we started going in, then found out that some of the materials we needed to run the studies and test, they're not available. Why? Well, supply chain issues. So that was frustrating. So now we roll into 2021. The supply chain is worse, but we're now sort of thinking, let's try to work together. Social distancing is being relaxed. Well, we still have masks, and okay, we figured it out. Then we found the devices we had made, which had been sitting on the shelf for over a year, did not have the same integrity that we had when we built them. We had initially planned on testing them within 3 to 4 months. And so when they started, they didn't have the same integrity. They have, for example, they had leaks. We called up the company and said, hey, why are they leaking? They said, well, it's after a year. We guaranteed them for 3 months. And yes, it may last a little longer, but if we can make them for much longer, you just have to pay more. But to give any resources, you own it for 3 months because you're going to test them right away. So we said, okay, we need to do that. Well, we found out that basically then we have to build the whole thing over again. So that delayed us. And that's why now we are sort of regrouping to get that work done. And it's pushed us back by a few years, but that's what the delay was. Liz, did I answer the question?
I think so. Yes. And there's a related question about funding. How much more funding do we estimate we'll need to get to the point of trials, and how much have we already raised?
So that's a great question. So when I put out this plan for how much do we need to get from the beginning of the, when we first came up with the cartoon to clinical trials, we are being supported by a grant from the NIH through a special program called the Quantum Grants Program. And they said, lay out your plan for how you get to clinical trials. We give a lot of thought, brought a lot of advisors, both from manufacturing and business. We came up with a number of $20 million from the time. And this was a plan we made in 2010 because the NIH wanted that. We laid out the milestones. And we raised close to 13, 14 million of that, but we never raised it at once. The NIH program, which was supposed to support all this, unfortunately got sunset. Basically means the program stopped without letting us complete. So then all of a sudden we have this plan. We've told people we need $20 million. We got some money, we couldn't finish. And people say, how much more do you need? We said, well, we'll still need 7 million, whatever the number was, 10 million, 7 to 10 million. And we'd never got it. We'd get maybe occasional grants, 100,000, 5,000, $50, appreciate all of it, $1 million. So what happens, we get the money comes in dribs and drabs, and we can't really do the work. Sometimes we take 1 step forward, 2 steps back. So if I was to look at today, how much money do we need? The number is probably on the order of, we had said initially $20 million. We raised close to $13, $14 million. We probably, again, if somebody wrote us a check today, I would say $9 to $10 million would take us through, or at least assure that would get it. We continue to raise a few 100,000 here and there, but we have not raised the millions that we need. That's the fundamental challenge. So to everybody out there, they're like, you always tell us $10 million. Well, if I got a significant chunk from that, I would say, okay, it's now $5 million, whatever that was. But if I get $50,000, I do some work, and then I have to wait for the next $50,000 to come in and spend another 2 months. It's almost like I go backwards. So we keep the project going. We are committed to the project. We do not want to shut it down. So we keep on going. We do everything. Maybe we throttle down. We say, okay, let's not go fast because we do not want to completely stop the project. But it turns out that number is there. So if anybody's looking at what is a sizable chunk, $1 million would make a significant dent. Smaller amount makes dents, and lets us advance it, but not in the same way that I have assurance if I have a large number.
Great, thank you for explaining that. And I think we're just about at the hour, but I'll pose a couple last questions from the chat to you. Would patients that are type 1 diabetics be eligible? And along with that, would patients that have IgA nephropathy also be eligible for iBAK?
Yeah. Again, I'll give you my opinion as a non-physician. And again, I think my physician colleagues would say, talk to your physician. But in principle, I'm assuming type 1 diabetics and IgA nephropathy, people who have kidney failure, not just the fact they have the disease. The answer is yes, possibly, although It will depend on your specific situation. There's no reason because you're type 1 diabetic, you would not be able to get it, or an IgA nephropathy. But it will depend on your condition. So the fact that you're diabetic, does it say our device will not work? No. But there may be other associated conditions that may make it harder. Maybe your blood vessels may not be the right condition to sew on our device. But again, that's individual. Maybe you have a drug regimen, if IgA nephropathy, that precludes from having a foreign material in your body. Now, it has adverse consequences. That again, in the individual case. But at a global level, the short answer is no. Those should not be disqualifying by themselves.
All right, that's good to hear. And the final question is, do you need more engineers working on the project, and what type of skills are needed?
We need all kinds of people, not just engineers. We'd love engineers. We'd love engineers who can design. We can love engineers who can machine. We can love engineers who can make silicon membranes. But we also want people with expertise in clinical trials. We want expertise in raising money. We want expertise in communication. Some of you may feel like I'm not an engineer, but hey, I am very good at communicating. Talk about our project. We need everybody who wants to contribute, because I believe we can deliver on this project together. It needs a team effort. We're not waiting for some new discoveries. We may make some, but we're not waiting on them. But engineers, scientists, business people, communicators, I can't think of anybody. I guess we don't need bad people, but we need everybody who's motivated.
Thank you. Well, I think that's a great place to leave it for this afternoon. Thank you all so much for tuning in today, and we will be back next month. And in the meantime, you can keep in touch with us with social media and our YouTube channel, and we look forward to being back again in October.
Thank you.